Cellular respiration, the process by which organisms combine oxygen with food submolecules, diverting the chemical energy in these substances in life-sustaining activities and discarding as waste products like carbon dioxide and water. Organisms that do not depend on oxygen degrade foodstuff in a process called fermentation. In this experiment, you will study the fermentation process of yeast and respiration rates of human, animal, and plant. After completing this exercise, you should be able to understand the fermentation process and effect of inhibitor on the fermentation and determine and compare the respiration rates of human, plant, and animal. We are going to start with fermentations of yeast. These are the chemical reagents and the equipment which contain with 5% of yeast suspensions in warm water, 20% sucrose solution. The prepared solution will be put into fermentation tube, 6 ml of yeast suspension, 9 ml of sucrose solution, and a few drops of methylene brew will mix together in the beaker before added into the fermentation tube, while another tube put water instead of sucrose. Then, put both tubes on the bready dish and leave them at room temperature. The gas in the tube was measured every 5 minutes for 30 minutes and also observe the color changes. After the experiment ended, add a pellet of potassium hydroxide. Shake it lightly until the pellet melts and mix with the suspension and the gas-filled upside of the tube and observe the gas volume. So, potassium hydroxide is a carbon dioxide solvent, which potassium hydroxide is a strong base, and carbon dioxide is an acidic oxide. So, the two readily react to form potassium carbonate, or salt, and water. That's why potassium hydroxide was used in this experiment. These pictures show the changes of gas volume and color in fermentation tubes. The gas occurred in the tube with sucrose, while there was no gas formed in the tubes with water. However, it's worth found that the colors of both tubes changed from blue-green to yellow. After 30 minutes, 2 to 3 pellets of potassium hydroxide were added into the tubes with sucrose. The results showed that the gas disappeared. This is the all results of this experiment. You should record on your lab report. So, gas volumes is in the unit of milliliter, and color was recorded by using positive and negative signs, which represent blue-green color and yellow color respectively. After you understand the fermentation process, we are going to study the effect of sodium fluoride on the yeast fermentation. Notice that glycolysis pathway is involved in fermentation process, and sodium fluoride is a potential inhibitor of glycolysis because it inhibits enolates, which is an enzyme midway along the glycolysis pathway. The method is similar with the previous one. For this experiment, there are 4 conditions as shown in this picture. The reagents were mixed in the beaker before put into the tube. All conditions contain 6 ml of yeast suspension, and the ratios of all the reagents vary as shown in this picture. The first tube was mixed with distilled water. The second tube was mixed with distilled water and sucrose solution. The third tube was mixed with distilled water, sodium fluoride solution, and sucrose solution. And the fourth one was the mixtures of sodium fluoride solution and sucrose solution. Yeast suspensions mixed with the other solution was prepared before added 2 to 3 drops of methylene blue. Then, the mixed solutions in the beaker was filled into the fermentation tubes, left at room temperature, and measure the gas volumes in the tubes every 5 minutes for 30 minutes. The amount of carbon dioxide gas was shown in this table. You should record and discuss this result on your lab report. You will see that carbon dioxide gas appear in all tubes, which tube number 2 with sucrose had the highest carbon dioxide production. Carbon dioxide gas decreased when sodium fluoride solution was added in the tubes number 3 and 4. We have already finished the fermentation process and now move to the next experiment, determinations of the respiration rate. Human, mouse, and germinating bean seeds were used in this experiment. For humans, we divide into 2 conditions: before exercise and after exercise for 5 minutes. This is the principles of this experiment. The carbon dioxide from our breath will dissolve in the water and becomes carbonic acid like this equation. Titration with base solution, sodium hydroxide, reacts with carbonic acid to form sodium bicarbonate, which is an alkali salt. Acknowledge that phenolphthalein is often used as the indicator in acid-base tritations. For this application, it turns colorless in acidic solutions and pink in base solutions. Put 100 milliliters of phenolphthalein solution in the flask. Notice the color. The solution color is pink, so is the solution acid or base? Blow the air bubble under the solution until it becomes colorless. Blow for another 1 minute. Titrate the solution with 0.04% sodium hydroxide solution until the solution turns light pink. Then Stop the titration. The color at the end point will not fade when leave it longer than 1 minute. Record the volumes of sodium hydroxide used in the titration and calculate the amount of carbon dioxide. Repeat the experiment again after you exercise for 5 minutes. Use new phenolphthalein solution and compare the amounts of carbon dioxide that you produce before and after exercise. Set up 4 of 250 ml flasks for the experiment. Put 150 ml of saturated calcium hydroxide solution in flask number 1 and number 2. Because the solution can be effectively used as an absorbent to capture carbon dioxide gas, it means that the amounts of carbon dioxide in flask number 4 will be generated from organisms. Then put amounts or generating bean seeds in flask number 3 and put 100 ml of phenolphthalein solution in flask number 4. Remove the air from flask number 4 using a water suction pump. The outside air will go through flask number 1, 2, 3, and 4 respectively. Make sure that there are air bubbles in every flask. Wait until the phenolphthalein color disappears, and then let the air pass through the solution for 1 minute. Titrate the solution from flask number 4 with 0.04% sodium hydroxide solution until the pink color returns and does not disappear within 1 minute. Record the volume of sodium hydroxide solution used. These results can be used to calculate the amount of carbon dioxide production from each organism. It was calculated in the units of micromoles that produced in 1 hour per weight using the following equation. The amounts of carbon dioxide equals to amounts of sodium hydroxide solution multiplied by 10 and multiplied by time and divided by weight as a gram. Heat is generated from respiration process. In this experiment, we are going to determine the heat generation from respirations of germinating bean seeds, cooked bean seeds, yeast suspended in sucrose solution, and distilled water. The experiment was set up in the thermos bottle like this and leave at room temperatures for 2 hours. Record room temperature and the temperatures of each thermos at the beginning and the end of experiment. The results were shown in this table. You will see that temperature from germinating bean seeds and yeast bottles increased, while that of cooked bean seeds was decreased. However, the temperature was stable in distilled water bottle. Record and discuss the questions on your lab report.